Here are two high-yield USMLE-style questions focused on molecular cloning and its applications. Question 1. A 32-year-old woman with diabetes mellitus receives treatment with recombinant human insulin produced through molecular cloning. Which of the following best describes the basic process used to produce recombinant human insulin? A. Insulin is synthesized directly in human cells using a viral vector for gene delivery. B. Human insulin gene is inserted into bacterial plasmids, which are then used to produce insulin in bacteria. C. Insulin is obtained from human pancreatic cells grown in culture and purified for use. D. Insulin is synthesized from a yeast culture that carries the insulin gene on its chromosome. E. Insulin is synthesized in vitro using synthetic mRNA templates. Answer B. Human insulin gene is inserted into bacterial plasmids, which are then used to produce insulin in bacteria. Explanation. Molecular cloning involves inserting a gene of interest, such as the human insulin gene, into a bacterial plasmid. The plasmid is introduced into bacterial cells, where the bacteria use the gene to produce the protein, in this case, insulin. The insulin is then harvested and purified for medical use. This method is efficient and widely used for producing human proteins in bacteria. Question 2. A researcher is attempting to produce a recombinant DNA molecule using molecular cloning. After inserting the gene of interest into a plasmid vector, the plasmid is introduced into Escherichia coli cells. Which of the following steps is most critical for selecting E. coli cells that have successfully incorporated the recombinant plasmid? A. Performing a polymerase chain reaction, PCR, to amplify the gene of interest. B. Inserting a gene for antibiotic resistance into the plasmid to select for transformed cells. C. Growing cells in a nutrient-rich medium to allow for protein expression. D. Using an electric pulse to introduce the plasmid into E. coli via electroporation. E. Treating cells with a bacterial virus to facilitate plasmid uptake. Answer: B. Inserting a gene for antibiotic resistance into the plasmid to select for transformed cells. Explanation: in molecular cloning, a gene for antibiotic resistance is often inserted into the plasmid vector alongside the gene of interest. After introducing the recombinant plasmid into E. coli cells, the cells are grown in a medium containing an antibiotic. Only the cells that have successfully incorporated the plasmid, which carries the antibiotic resistance gene, will survive and proliferate, allowing the researcher to select the desired cells for further analysis or protein production.